Before we get started, we would like to provide you with some key concepts that are used in this lab. For example, photosynthesis is a process used by all plants. It converts carbon dioxide into sugars such as glucose using energy from the sun. Photosynthesis uses a process known as carbon fixation, which uses pigments in a leaf to absorb light energy in order to convert carbon dioxide into glucose.
Now let's get back to our lab. The purpose of our lab is to observe the rate of photosynthesis under different lighting conditions, including low light, ambient light, and high-intensity light. Here are a list of materials that we will use in our experiment. One of our materials we will be using is a hole puncher. As you can see below the hole puncher, we have spinach leaves. There is no set number of spinach leaves you will need. You will just need enough to have 60 discs punched out by a hole puncher. Or, instead of a hole puncher, you can also use a straw. Next, you'll see the 6 10-milliliter syringes in the center. These syringes will contain the disc that we punched out from the hole puncher. There should be 10 discs within each syringe. Finally, to the left, you will see that we have 15 milliliters of sodium bicarbonate and 15 milliliters of water. 3 syringes will contain the sodium bicarbonate and 3 other syringes will contain the water. All of them will end up having 5 milliliters of liquid each. We also can't forget about our environment. For low light, we will be using draws. For ambient light, we will be using the table. And for high intensity light, we will be using this high intensity lamp. In fact, the intensity of light will also be an independent variable. This includes the low light, the ambient light, and the high intensity light. The dependent variable are the number of leaves floating in the syringe, which ultimately indicate the rate of photosynthesis. Experimental groups are the groups receiving high intensity light and no light, while the control group is the group that received the ambient light. Our problem that we will be exploring in this experiment is which kind of light will produce the fastest rate of photosynthesis. You can use your pre-existing knowledge of photosynthesis, factors affecting photosynthesis, and respiration to hypothesize what you think will happen in this experiment. Hint: the sodium bicarbonate increases the amount of carbon dioxide dissolved in water.
After gathering all the materials needed, the next step will be to cut out 60 spinach leaf discs with your hole puncher.
To ensure that your research is uniform, make sure that all your leaf discs are similar in texture and thickness while avoiding major leaf veins.
Then, pull out the plunger from the syringes and carefully place 10 discs into each one, being sure to place the plunger back in the syringes.
You should end up having 6 syringes with 10 discs in each. 3 of them will be reserved for water, and the other 3 will be reserved for sodium bicarbonate. Now you will fill 3 syringes with 5 milliliters of water, and the other 3 syringes will be filled with 5 milliliters of sodium bicarbonate. I'm sure all of you do chores in your house. How well are your vacuuming skills? Well, it's time to put them to the test. Except you're using a syringe. But not to worry, we'll show you how. First, you should push the plunger, removing as much air as possible from the syringe. Then, you will hold a finger over the syringe opening and draw back on the plunger to create a vacuum simultaneously. Hold this for 10 seconds and swirl the syringe so you could get all of the leaf discs suspended in the solution. Then, you will slowly let out the vacuum and repeat it 2 or 3 times until all leaf discs sink. However, you should be careful not to yank off the plunger because, yes, it can spill. And also, you could damage your leaf disc. As you can see now, I'm starting to tap out the air bubbles. Be patient and wait for your disc to fall. Again, repeat if necessary.
Tap on the syringe again to get rid of those pesky air bubbles. You should see all the leaf discs start to sink. Repeat this process until all the discs sink. This is what they should look like when you are finished.
Great job, guys! Now you will have to place 2 syringes in each environment. There will be 2 in low light, one of which will be water and the other which will be sodium bicarbonate. 2 in ambient light, again 1 water and the other sodium bicarbonate, and then again 2 in the high intensity light. Now you should visit your syringes every 2 minutes to see how many discs have now floated.
What you should observe while performing this experiment is how many leaf discs will start to float. Why does this occur, you may wonder? This is a result of photosynthesis and oxygen being produced as a result of photosynthesis. In other words, the syringes that had the most amount of leaf discs floating had a higher rate of photosynthesis, so more oxygen was being produced.
The following is an example of a table you can use to organize your data. Let us know in the comments below what the results of your experiment were and whether your hypothesis was falsified. Thank you all for watching. Please leave a thumbs up and subscribe. Oh, and if you're still here, enjoy this video of our guest star, Henrik.